Good afternoon to everyone. We are working on the biggest problem of life, whether in earth or space, and that is death. And I'm not a politician, I'm not campaigning here. I'm talking about cancer. So I'm the founder and CEO of Genevation, and we are working on cancer. How to solve cancer effectively, efficiently, and in a personal way. The biggest challenge is not that we do not have treatment options, but we do have treatment options, but they are slow. They're not personalized. They are toxic, it's painful, and it's cost effective. And when I am talking about death, the number is massive. So every year, around 20 million people across the globe are affected with cancer. 20 million is a big number. And the death rate is strikingly high, 50%. So we are talking here about numbers, 10 million. We are losing every year 10 million people dying of cancer. And we do not have an efficient treatment. So what are the things we are lacking? So what we need is, we need precision treatment because, as I said, cancer is very personal, but we have generalized treatment. We need non-toxic approaches. Go and ask somebody who has gone through chemo and radiotherapy. Ask them how their life changes before and after cancer, before and after treatment. You can talk about grade 1, grade 2, grade 3, grade 4, doesn't matter. The message is very simple, very clear. We do not have a safe treatment, and we do not have a treatment which is not painful. So what do we need? We need improved outcomes. Because we are stuck with big pharmas, we are stuck with investors, we are stuck with management and board and all those fascinating things what you see with big pharmas. And it also has to be affordable. Because cancer, there are many types of cancer which is so costly in anywhere of the world. Whether the government pays or you pay, doesn't matter. These are very costly treatments. So do we have an option? This is the problem I have spoken about. So what are the options we have? Now let's look at the options we are talking about. So we have found a end-to-end solution for that. So we have developed a vaccine, personalized vaccine for lungs, colon, and skin cancer, preclinical. We have tested those vaccines in mouse model, although it's animal because we can't test in humans, unfortunately. And we have developed that end-to-end in 14 days. So what happens? We take a biopsy, we do a sequencing, which we have completely disrupted. We look for the best new antigens or vaccine candidates. We have our own algorithm, which we select them efficiently. Then we coupled with machine learning model. We then have a synthetic vaccine manufacturing platform. And then we dose the mice, in this case for the patient. So the whole end-to-end takes only 14 days. And I think we are the first company globally who have done a preclinical trial where we make a vaccine in 14 days. And what I say, what do you hear? So how do you integrate that? What are the things, what do we use and how we use AI? So this is a platform, it's not just on AI, but the whole algorithm which we use for sequencing, for facilitating the process. So basically the traditional approach, it takes 3 weeks time. So using our algorithm, using our unique bioinformatics, using our unique sequencing, we then coupled with AI, which we takes 1.5 days. So we are saving a massive time. Instead of 3 weeks, we are generating a platform means where we are identifying new antigens which are cancer specific for those individual patients efficiently, and that takes just 1.5 days. So the next question is, how do you know that we have generated superior neoantigens or the vaccine candidates are superior? The obvious thing is to compare. So we compared with the best companies which are well known for making vaccines, and we compared with the preclinical data which they had in mice and with us. And we have got a remarkable success. So 95% of our neoantigens or cancer vaccine candidates in layman's terms are highly effective. And those effectiveness I'm talking in the platform here. What I show here is, I'll come to the next slide and I'll prove it, the validation, which is the most important part. So what we have done is, once we have generated the vaccine candidates, we have compared them side by side with all the known neoantigens preclinically available. And we have seen 95% of our cancer vaccine candidates are so effective in causing an immunity or immune response in mice. Compared to the big companies, big pharmas, which they are just 1% of the candidates have been shown effectiveness in mice. And as I said, this all looks good in PowerPoint, fabulous, great. So we need validation. That's where the real science comes from. It's not about the data. It's not about the fact that we are using something to speed up efficiency. The efficiency only matters when the quality matters, when you get an immune response. So let's look at. So this is one use case of lung cancer. So we have done two types of vaccine. One is prophylactic as well as therapeutic. So this is a massive, like with a one suboptimal dose, because we have a different dosing system, we have a different delivery mechanism, we are non-LNP based, we don't do the usual way which the vaccines are made. We completely disrupted the technology. So here you can see in the control, the blue one, compared to the red one, which has been vaccinated with the neoantigens. Until 11 days, the tumor was completely wiped off, or the tumor was suppressed. It only came back on the second dose, and this is because we are using suboptimal dose, but still the immune response was at least 54% until the day 29. So what we did a therapeutic vaccine trial in mice. So the blue one is the control one, the red one is the vaccinated ones, which had the neoantigens. So what we did was, since, as I said, that we use suboptimal dose, this is just 2 doses compared to 10 doses with other pharmas or other preclinical trials. I asked my data scientist to mimic and predict using the real data points how many doses will be required. Suppose if you take the standard 50 microgram. So you can see in brown and the darker ones, in 4 doses using 50 micrograms, we could get rid of most of the tumor, whereas with 8 doses, we are completely abolishing. So that is for the therapeutic vaccines, like once you have a tumor, and then you vaccinate the patients. What about the prophylactic ones? So similarly, we have chosen the prophylactic vaccine candidates. We have vaccinated the mice. And what you see in the blue is the control. The green is that what you got the real-time data, and the lower below ones are with 4 doses and 8 doses with 50 micrograms. And again, I say this is predictive model. In theory, it would work much beneficially because in humans, we can dose more than twice. But unfortunately, due to our pattern of dosing, the FDA guidelines are very strict. We cannot use more than 2 doses, and that too suboptimal doses. And the next thing what we did was we wanted to know how accurate the predictive dosing models are, because it's very easy to predict anything. So these are the lines, the dots are, which is observed, the real-time data, and the lines, what you see, the straight line is the predictive model. So you see that we have nearly 100% efficacy or efficiency, the graph shows, in predicting the data. So why do you think we win? So because we are not only predicting, but we are using those vaccines, we are testing in mice, which is validating our own data. We got a in vivo validation mechanism, and our R&D platform is building a strong data based on our own. We are not collecting data from any clinics or any universities or anyone. We are just using our own data because we have a complete different and disruptive platform technology. We have an IP, which is a global PCT application pending on this one, on therapeutic vaccine and prophylactic vaccine, which is synthetic platform. We have a trade secret to identify the most efficient vaccine candidates. And we use our own data, preclinical, and we look for the immune responses, which are in-house, and which is built for predicting neoantigen. And currently we are doing in lungs, colon, and skin, but we'll do across multiple cancer types, even some other infectious disease also. That's the plan. And we could generate revenue with licensing with pharmas. That is another option. And we are one of the first company to make end-to-end vaccines. Like we are making our own neoantigen, and we are vaccinating it. So we are first end-to-end companies who are not using CDMOs or CROs to either predict or to manufacture vaccines. So what is next? So now we have done successfully the preclinical trial. We are raising $5 million for IND. We are going for IND submission, and we are expanding the platform with different cancer modalities, infectious disease, because the US government is very keen on our faster vaccine platform. So we are working closely with the US government on the infectious disease like Ebola or influenza. So there is go. And this is all about the team. This is myself. We got Andrew, who is our CFO, which is an experienced CFO. We have Sandeep, who is the SDSU director, who is a doctor, he is very experienced in immunotherapy clinical trials. And we got a fabulous guy, Stephen Cheng, who is an NIH chair and a study director as our scientific advisor. Thank you, everyone. Happy to take questions. Thank you.